glad you're doing a project on chemistry, and especially organic chemistry, so that's fantastic.
Who gave you this assignment? See, a chemist gave you this assignment, right? Not a biochemist. Huh?
Yep.
So I'm an organic chemist by training. I got my PhD in France. I did a postdoc in England in School of Pharmacy, where I prepared anti-malarial compounds and tested them. Mm-hmm. Then I moved to Georgia Tech as a postdoc in chemical engineering, working on new solvents, really. Okay. And then I worked in industry for a little bit, in the pharmaceutical industry up north in Delaware. And then I came back as a research faculty, and now I'm a general faculty at Georgia Tech. So my research used to be, to begin with, on smart solvents, so very heavily on sustainable chemistry, green chemistry, and engineering. So we developed a sustainable solvent for efficient separation after reactions. I worked on polymers. I worked on, what did I work on? So mostly sustainable on pharmaceutical processes using continuous flow technology. Also, the goal is also green sustainable processes for high value added compounds. Okay. And recently I've been, in parallel to the experimental research, I've researched in chemical education as well as chemical safety. Okay. So that's kind of a background.
Yeah, so with like all the sustainable...
Sure. So I received my bachelor's degree in chemistry from the Georgia Institute of Technology in 2014. And I went to graduate school at Massachusetts Institute of Technology, where I defended my PhD in 2018, and became a faculty member at Salisbury University immediately after that, and now I'm a faculty member at my alma mater, Georgia Tech. Yeah. My undergraduate research focused on developing palladium-mediated reactions to make materials, organic materials for things like light-emitting diodes and solar cells. And then my graduate work at MIT focused on developing new palladium-mediated bioconjugation reactions, specifically at modifying the cysteine amino acid of biological molecules like peptides or proteins.
So I'm Angus Wilkinson, been a professor at Georgia Tech for 30 years. I'm an inorganic materials chemist, so in fact principally work on ceramics with controlled thermal expansion and also cement systems. I have a collaborator over in civil engineering where we're working on developing what are called low embodied CO2 cement. So these are basically cement systems that have a lower environmental impact than standard construction cements.
I teach biochemistry.
So just Biochem 1?
Or? Yeah, so the 32, it used to be, they just changed the number. It's like 3281 or something. But it's the first semester biochemistry.
Oh. Okay.
I teach that. I'm not the only one who teaches it. Other people teach it too. Mm-hmm. But I teach that, and I also teach some graduate level classes. But pretty regularly I teach biochemistry.
Oh.
Yeah, absolutely. So, the beauty of biologics is that it has had millennia, if not millions of years, to evolve over time to become really good at specific functions in a way that on a human lifetime scale isn't possible to do. And because of this, everything that a biological molecule does, it does so incredibly energetically efficient and very specific to specific targets. And because of that, a lot of researchers seek to mimic all of the reactivity that biological molecules can do towards new things. So for and a simple example might be, your body digests some food that you consume, and there are different enzymes in your body that specifically convert one type of molecule into another one. And it does so incredibly efficiently and with an incredible degree of specificity. And the specificity is probably the most important part, because swimming around in the food that you consume are billions of types of molecules, yet the enzymes in your body will specifically target just a single one. So another example of how we might use biologically designed molecules is in the area of therapeutics. So developing new targeted drugs to treat some disease. So for example, we can take a peptide that is designed by humans and use it to inhibit different protein-protein interactions inside of a human body to prevent or to eradicate or to treat some other disease. And this is a common thing that is done in therapeutic design. For example, probably the most famous recent example, in fact, just today, is for a vaccine to treat COVID-19.
When I was much younger, before I came to Georgia Tech, I was at a location where they were working on abalone shells, which is actually an inorganic organic composite. So these shells derive their extreme toughness by virtue of combining biologically derived inorganic polymer with crystals of calcium carbonate, and actually different polymorphic forms, so different structures at the atomic level of calcium carbonate as you move through the shell. So you have something that looks like kind of a brick wall where the bricks are crystals of calcium carbonate and the organic material is like the mortar in the brick wall. And that actually gives the shell toughness.
Well, that's a pretty broad question. So aromatic phenol is the most simple aromatic you can find, right? So aromatic compounds are characteristics. So I didn't tell you that, but I do teach organic chemistry for a while. So aromatic compounds, I don't know how much you study the structure, but aromatic compounds, the term aromaticity really just means an unusual stability. And benzene is really the most simple one that you can find. So it needs to meet certain rules to do that. It needs to be planar. It needs to have a continuous array of p orbital, and so that's where you have your pi system and your pi electron are being shared, and that's what you may have seen as a pi cloud. But you also need, once those three are met, so it needs to be a ring, of course, but it needs to be planar and have this continuous array of p orbital. To really be aromatic, you also need to have 4n plus 2 pi electron, which means either 2, 6, 10, etc. And so this extra stability has been observed experimentally for a long time, although the understanding of the aromaticity came much later. And so those compounds, because of their extra or unusual stability, have been present in, as you said, natural compound as well as in synthetic compound. So the beauty of aromatics, like benzenes, they can also interact through their pi system. So I don't know how much you have worked with intermolecular forces. So compounds, boiling point, for example, so physical property like boiling point, are affected by the intermolecular forces. So those are the interaction from molecule to molecule. And so the one that is the most well discussed probably is hydrogen bonding. Benzene itself is carcinogen, so we moved away from it. But it has been used as a solvent because of its solvation properties. Okay. So in natural compound, it brings you this kind of balance of polar, non-polar. You cannot be only polar because your cells have some non-polar sides, right? Mm-hmm. So it gives a pretty good balance, and its planarity is really important in its ability to go through systems.
Okay. So
a phenyl group is a 6-membered carbon-hydrogen ring that contains 6 pi electrons, mm-hmm. and in this phenyl group, this phenyl group is found in nature in biological compounds, specifically at a few different sites. So one of those sites is going to be the amino acid tyrosine, has the phenyl group in it. Mm-hmm. And several other compounds as well have phenyl groups in it. And these phenyl groups are capable of being modified at different locations, and while maintaining a lot of the same properties that the unmodified phenyl group has. So it's really tolerant of modification without disrupting some of the interactivity between other molecules. And an important part of that comes from the pi-to-pi interactions. So these pi-pi interactions, it's spelled P-I-P-I, by the way, mm-hmm. Allow for the stacking and intermolecular attraction between different molecules. And one way that researchers harness this, specifically for biological molecules, is it basically allows molecules to bind together to some degree without falling apart, because these pi-pi stacking causes an attractive force that keeps those molecules together. So, for example, some people, in fact, have used this to help direct modification. So if they can create what's called a pi clamp, if you look up pi clamp, you'll find information about this, where they take the design principles of pi-pi stacking and allow it to direct modifications at molecules through this pi stacking interaction. And again, if you look at a phenyl group, you'll see the 6-membered hexagon, but inside there you'll see 3 lines, and those are indicating a double bond. And those double bonds are what are called pi electrons. And those pi electrons, there are 6 of them inside.
Okay, one thing about a phenyl group is it absorbs light. Aromatic compounds, like phenyl group is aromatic, and it absorbs light, and that makes it very useful. Like when we purify proteins and we want to know how much we have, we put it in a spectrometer and we look at the absorbance of the aromatic molecules. So that's very useful. That's one of the things about phenyl groups, is they're a little kind of optical label, so we can find things. If you look at most amino acids like alanine and other things, they don't absorb light. And so phenylalanine absorbs light, and that's very useful. So yeah, it's stable. A phenyl group is stable, it's aromatic, it's planar, it's greasy, it's not soluble in water. If you look at proteins, the phenyl groups are all on the inside, because the hydrophobic effect, pushes all the hydrophobic, a phenyl group is hydrophobic, mm-hmm. So it's not soluble in water. And so on proteins, it's on the inside, whereas the hydrophilic things, have you learned about the hydrophobic effect in, you probably haven't.
Yes, hydrophilic.
Oil is not soluble in water.
Right.
Yeah, okay, so a phenyl group is hydrophobic, and so it's one of the things that drives protein folding, because it goes on the middle. It comes out of water, so the middle of a protein is out of solution, and so phenyl groups drive protein folding.
Oh, so this is
So
yeah. Would you say that it kind of is in the tertiary structure of a protein?
Yes, exactly. And it's on the inside.
Okay.
So yes, it's on the inside of phenyl groups tend to be on the inside of proteins, and they're big, and they're rigid kind of, and and they're really important in protein folding, getting to exactly the right state, enzymes and things, they have to be in like just this right conformational state.
Oh,
and the phenyl groups are very important in that.
So when you say protein folding, are you talking about like local folding, so like secondary, so like alpha helixes and beta sheets, or is it more like three-dimensional folding?
Three-dimensional, yeah. So an alpha helix, they, like if you look at hemoglobin, it has a bunch of alpha helices all folded up, right?
Right.
And so that's called the tertiary structure, like you said, and that's in that tertiary structure, all the phenyl groups are on the middle.
Okay.
On the inside, they're not exposed to water because they're not soluble in water. So yeah, you have to look at the tertiary structure. If you just look at an alpha helix all by itself, every amino acid is exposed to water if you just have an isolated alpha helix. So in a folded protein, the inside, the alpha helices come together, and so there's parts that are not exposed to water. That's at the tertiary level.
So a lot of the aromatics, benzene-based compound, have been used in fragrances for a lot of time. They also used to be called paraffins because of their extra stability due to the aromaticity. But the reason why they are called aromatics comes from the term aroma, which is the smell. So before the understanding of their properties, they noticed that the compounds that add phenols and functional groups on it will add aroma associated with it. So if you look at a lot of, I think, so you have vanilla, you have the banana flavor, you have the strawberry flavor, the pear ones, they all have an aromatic ring. And so this could be one of the examples that a lot of the artificial flavoring are based on synthesis around the aromatic ring, like the phenyl.
Oh, well, okay. Let me think about that. I sort of look for, I'm sort of more of a biologist. I actually look in biology for what, honestly, let me think about how to answer this. Yeah, okay, let me just tell you sort of my perspective. If you look at biological molecules, like polymers, like protein and DNA and or carbohydrate and things like that, those are like biological polymers. And they're kind of like magic. Well, no, they're not like magic, but they're indistinguishable from it. Have you ever heard of Arthur C. Clarke's law of technology? He says that any sufficiently mature technology is indistinguishable from magic. Meaning, like, let's say it was 1950 and you had an iPhone and it was working, right? It would be like magic, right?
Yeah.
And so biological molecules are essentially indistinguishable from magic in that they have properties that are so incredible and so amazing that we can't really understand them. We kind of mimic them sometimes, but if you look at the properties, like protein, for example, which has phenyl groups, phenylalanine and things like that. Protein can be an enzyme. There's so many different fibers that are made, spider webs and hair and tendons, all these different fibers. And then there's adhesives, there's pumps and channels and motors. Just the things that protein can do are so beyond human comprehension that the biological molecules are, I'd say, indistinguishable from magic. And so for me personally, I am trying to understand where did these come from and how do they work, and really trying to understand them on a basic level. And then to say that, so I would say we can use them, but we don't really understand them. They don't, I don't know. I'm not helping you very much, I can tell that. I just think people don't we can say we're using biologically inspired stuff, but we're really not, because we really don't understand it. It's like too far beyond us. Like really, what from biology, what technology have we really taken from biology?
Well, maybe it's
Yeah,
something to do with the structure of, like, maybe a phenyl compound, and that structure is maybe used in the pharmaceutical because of maybe its stability or something to do with that.
Yeah, but that's just chemistry, though. That's not really that's not bio-inspired, is it? I mean, phenyl groups, they're hydrophobic, they stick to stuff, they, okay, I don't know. Like, okay, to give me an example, really, I wouldn't call that okay, one of the things about biology, that you have to look at is really, it's about polymers. It's like functional groups, like a phenyl group. Like a phenyl group all by itself is like, I don't know. But once you put it into a coated polymer like protein, and you put phenylalanine and things in the right place relative to other things, that's when it becomes. I'm not helping you, am I? I can see this.
Well,
Okay,
I kind of
help you.
get this contrarian viewpoint that there is this notion that we are biologically being inspired, but really we're not, because we don't understand the biology and the chemistry behind it. We understand how they work, but we don't know how to apply it. Is that kind of where you're coming from?
Yeah. Yes, and I'll say this: chemistry. I mean, I'm trained as a chemist, and the sort of the philosophy of chemistry is simplicity. We like to work at one thing at a time. You have to, and in order to publish a paper, you have to crystallize it and do CNH analysis and NMR. You have to have that like pure one thing.
Right.
But if you look at biology, it's never like that, right? It's always mixtures of things. It's always molecules interacting. And I mean, we don't, it's like, I guess it's like chemistry is like a cornfield in Iowa, and biology is like the Amazon jungle. It's just at a higher level of complexity. And chemistry, I mean, we steal some things from biology, but not very much.
Mm-hmm.
Not very much. It's like over our heads. Okay, but this is not the answer you wanted, is it? Okay, so let me. Yeah, and one thing about biology, it doesn't really want stability. Biology does not. Like if you look at all biological molecules, they're all unstable. I mean, proteins misfold. That's what amyloids are. And proteins are just barely. That's why if your body temperature goes up just a little bit, everything breaks, right? I mean, it's not. Biology does not try to get epoxy cement and things like that, right? It just, everything in biology is just nominally functional, and it's unstable. Okay, first you gotta define stability very carefully. You haven't taken physical chemistry yet, but stability is a thermodynamic notion, and thermodynamics means what happens after infinite time. Equilibrium. If you have something and you leave it for infinite time, it reaches equilibrium.
Mm-hmm.
Right? So if you take a protein and you put it in the Pacific Ocean for infinite time or anywhere, it will hydrolyze into amino acids.
Mm-hmm.
So proteins are unstable. They're kinetically trapped, meaning they hydrolyze very slowly, but proteins are not stable. Nothing in biology is stable. Everything is going to fall apart. It's just a matter of how long it's going to take.
Okay.
So the idea that biology teaches us about stability is not really correct.
Okay.
I'm like doing a terrible job of helping you here. Okay, I'm going to really try harder from now on.
So you have to be a little careful with toxicity. So the benzene itself, C6H6, the compound, is a non-carcinogen. Now you have a ton of compounds that will have the phenyl group and have no toxicity whatsoever. The flavors, the natural flavors, for example, is one of them. Mm-hmm. So toxicity is a complex toxic behavior is a complex thing to understand. You can see, for example, when you have the benzene, I don't know. So benzene is this, right? Where you have hydrogen at each carbon here. And so that's why I mentioned to you, it's planar, and every carbon is what we call sp2 hybridized. I don't know if you have worked on that.
Yes.
So benzene, C6H6, it's a non-carcinogen. Now you will put something like very similar. Now you replace one H by a CH3. And so this molecule is called toluene. Okay. And this is not carcinogen. And you didn't change much, right? You just changed one H by a CH3.
Right.
You can use phenol, that has been used for a lot of things. And phenol has some toxicity, but is not as carcinogenic as C6H6, for example.
Okay.
So carcinogenicity, it's a little tricky. You have to be careful with it. But it comes from the fact that it can intercalate, and it comes from the fact also that benzene has a very low boiling point for an aromatic, and one of the reasons it has not a lot of intermolecular forces besides the pi-pi, so it's pretty volatile, and so its inhalation can be quite dramatic.
Okay. And a final.
Yeah. So one of the major benefits of a phenyl group is how stable it is. So basically, it's not susceptible to oxidation. It's not susceptible to protein degradation. And because of that, it makes a really stable interaction and a really stable functional group to have as a part of your molecule. Basically, nothing can mess it up. But conversely, there are times when you would want a less stable thing to be present. So, for example, if you're interested in developing antibody-drug conjugates, so an antibody-drug conjugate, the way that it works is you have the antibody that is protein-specific that will bind to certain targets in a human body, for example, and it also is attached to a drug. And this antibody is what is responsible for directing the drug to go to the right place in the human body, and then presumably the drug will fall off and do whatever treatment it's supposed to be doing. However, if it is attached via a phenyl group, because that phenyl group is so stable, it's not going to fall off, and then all you have is an accumulation of a drug somewhere that's unable to be released into the part of the body that it needs to be released in to go do its job. So as you can see, it's kind of a double-edged sword, right? Where you have all of these benefits, but at the same time, sometimes those benefits can be drawbacks.
Well, they have poor solubility. One of the things you need, like when the pharmaceutical company is designing drugs, small molecule drugs, they have to be soluble in water, right? And phenyl groups are not soluble in water. So if you put phenyl groups onto things, they will fall out of solution, and that's bad. That's not something you want in drug development. So, there is I don't know. I mean, are phenyl groups good for anything? That's what you're asking me. I mean, they're...
Wow. It's almost ubiquitous to organic chemistry, the phenyl group. I think they are... It's almost everything. Now, there is a lot of variation on the aromatic, like phenol. You will have systems that, a lot of biological systems will have things like... So this one is, that I don't tell you something wrong, I think this one is a pyrimidine, which is also an aromatic, but as nitrogen instead of the carbon, right? You have pyridines that has one. So those are used, for example, so this is very important for biological application. You have the pyrimidine, you have the pyrazine, that is the nitrogen are in opposite positions. And so you have a lot of those. So this is pyrazine. And then you have pyridine that has been used also quite a bit in conjugated polymers, because you can use the advantage of introducing nitrogen because of their increased polarity, so you have more water solubility, for example. So for biological compounds, that's very important, since everything in biology is water soluble. So I think there will be more and more things going away from the benzene itself, but going to more what we call heteroaromatic. So those that have only carbon, right, are called aromatics. When you replace some of the carbon by nitrogen or oxygen or any more polar, it's called heteroaromatic. And so this kind of enhanced the water solubility while maintaining the aromatic characters that the phenol may have. So I think those are going to be important more and more as we see that we are trying to mimic biology, but we also are trying to have molecule or catalysts that do reaction in water, which means that not organic solvent, which is very different, and also at room temperature, where most of the organic synthesis tends to be at elevated temperature. So as we try to mimic nature, try to go back at systems, those heteroaromatic, I think, will become more and more important.
That's a great question. I think incorporating it into more proteins is going to be a large area of research. So it is an unnatural occurrence, the way that me and other researchers have incorporated it into the human body. So what that means is that when it is identified as a foreigner to a human enzyme, for example, the body is going to do whatever is possible to get rid of it. So figuring out ways to kind of like sneak it under the radar is important. And then in addition, I think, developing new payloads that can be released. So as I just mentioned, one of the drawbacks is that it is incredibly stable, so it often won't release whatever drugs are being attached to it. Finding ways to kind of leave it on and make it stable enough so that it reaches its target, but then once it gets there, how do we get it to release, given that it is bound via a really stable interaction?
Okay, in there's this thing called molecular recognition. How do molecules recognize each other? When you have a drug, most drugs are inhibitors of enzymes, and they bind right in the active site in a very specific way. It's called molecular recognition, right? They have to recognize that active site.
Right.
And one of the ways that is done is with what's called, there's a bunch, so there's things called hydrogen bonds, there's dipolar interactions, there's a whole bunch of things that are involved in molecular recognition.
Mm-hmm.
And one of them is called cation-pi interactions.
Okay.
And that's when you have a pi system, like a phenyl ring has a pi system, and a cation, like a lysine group or something, will sit right on top of it.
Okay.
And that can be involved in recognition, which is used in drug design. It's called cation-pi interactions. Okay. That is the answer that you need. So if you Google cation-pi interactions and drug design, you will find a bunch of stuff.
Okay. Yeah, I'll definitely look into that.
Yes. That's what you want, cation-pi, because that's the phenyl. See, the phenyl group is an aromatic system. That means it's a pi system, right? It has sigma bonds and pi bonds. Mm-hmm. And the pi bonds, right? The pi bonds are the electrons sit above the ring. The sigma bonds, the bonds are right in the plane. Pi bonds, they're above. Okay. And so that's a kind of a pancake of electrons, which is negatively charged, and then a cation sits right above that. Okay. So the cation-pi interactions are very important in drug design. Yes. And that is also, cation pi interactions are also important sometimes in protein folding and things. So that is an example of bio-inspired design for sure. Cation pi interactions in drug design. There.
Okay.
You got it.
So now you believe in bio-inspired designs?
Yeah, I do.
Okay.
Yes. We have an example of it.
Okay. Yep.